Welcome back as we continue our tour of the cell, and at this time we're going to talk about the endomembrane system. So how we produce proteins and then either use them right in the cell or ship them out of the cell for use other ways. And that is part of the endomembrane system. So it's going to be the packaging, modification, and movement of materials, specifically proteins. And if we look at the diagram shown here, we can see on the left in the red is the nucleus. And we talked about how the mRNA may have to leave there to produce the proteins. And then just outside that we have the endoplasmic reticulum, the ER. And in this case it's rough ER because it has ribosomes that are going to produce the proteins. That's going to produce vesicles that will have the proteins in them. And in some cases, those vesicles may go directly to the cell membrane for export out, but in many cases, they'll go to the Golgi apparatus, which is shown in the blue, where there they'll be modified, repackaged into vesicles that then will leave the cell through the cell membrane. So we're going to look at each of these pieces of the system, the ER, the ribosomes, and the Golgi apparatus as we go through. So the first one we're going to look at is the endoplasmic reticulum, or the ER. Excuse me, and this is the highway of the cell. So this moves and processes proteins, and we can see it is kind of this just kind of network, and that's literally what it means, a small net just outside the nucleus. And in some cases there's ribosomes attached directly to it, which is rough ER. In other cases there's no ribosomes, so that's smooth ER. So let's look at those two separately briefly. So the first, rough ER, and these are going to be proteins for export. So here's where proteins are synthesized directly into the ER so they can be packaged, processed, and shipped out. So if we look at this lipid layer here, here is the endoplasmic reticulum, and there are translocon channels, they're protein channels on the surface of that where the ribosomes will attach. And in just a second we'll see how the ribosomes are made up of two subunits. But these ribosomes attach, they're going to read the mRNA from the nucleus, and as they synthesize the protein, they are putting it right into the space in the ER. So this black thing that looks like thread is the new polypeptide or protein, and it's being processed directly into the ER so that it can be packaged and shipped out of the cell. So there's what rough ER looks like. Smooth ER doesn't produce protein because it doesn't have the ribosomes attached. And the main function that I want you to kind of remember is that it contains enzymes that help detoxify lipid-soluble drugs. So it's involved in the detoxification of toxins in the body. And from there we're going to move on to the ribosomes. Now the ribosomes, as we can see here, are made up of two subunits. There's a small subunit and a large subunit. And when we talk about protein synthesis in our next unit, we'll get into more specifics. But they will click on to each other, and mRNA would feed right through the middle of this to be read to make that protein. So the ribosomes read the mRNA to produce proteins. And we can have either, as we've seen, they can be attached to the ER, in which case it's rough ER, or they can be free floating in the cytoplasm. And that's usually where proteins that are going to be used within the cell are made. So for now, you need to understand that there are attached ribosomes that are part of the ER, and then there's free floating ribosomes that will produce proteins that are going to be used directly inside the cell. Now the last part of the endomembrane system is the Golgi complex. And the Golgi complex kind of looks like this series of flattened pancakes, a stack of them. And there is a cis end, and they receive vesicles from the ER. So you can see right here. So those proteins that we talked about that were made directly into the ER, they are packaged. They'll float through the cytoplasm to the Golgi apparatus, and the cis face of them receives that. Here they're modified. Maybe they've got some carbohydrate added to them to become glycoproteins, so that will become part of the cell membrane, but they're modified. And as they kind of work their way through the Golgi apparatus, they reach the trans face. Here's where they're going to be repackaged into vesicles, and they are going to be dragged towards the cell membrane for release into the bloodstream, or sometimes they will be incorporated as new cell membrane. And so if we look here, here is kind of the transition from vesicles from the ER to the Golgi apparatus, where they're modified, repackaged into secretion vesicles. Those will travel through the cytoplasm to the cell membrane, where the contents can be released to the outside into the bloodstream. One other thing that you can happen, so what we saw was just kind of this bottom root with the pink dots. If we look at the top with the blue rods and the black, what can also happen is these black rods are part of the membrane, and when that fuses with the cell membrane, they are left there as glycoproteins, as membrane proteins that are used for cell recognition and such. So this is one way that the cell can grow, by adding new membrane through vesicles produced by the Golgi apparatus. So there in a nutshell is the endomembrane system. And I don't want to go ahead there. The endomembrane system and the rough ER, smooth ER, the Golgi apparatus, the vesicles, and the ribosomes related with that. So until next time, we'll see you then.